So here is another slide that we would show our year 6. So this is a content slide noting the different types of cells which pertains to the content that students must be exposed to and retain in order to meet the success criteria. So here we have listed the different types of cells such as blood cells, brain cells, skin cells, liver cells, bone cells. So the rationale for this content slide is the inclusion of multiple different types of cells allows students to think about cells broadly and the many different examples of cells. HITS, as in the high impact teaching strategies, calls this teaching strategy multiple exposures. It relates to the chosen curriculum, as it states that students are to be exposed to living things, as in multiple living things that have different structure features and adaptations to survive. So an example of this is skin cells in humans that are found in hotter climates. So humans who live in hotter climates, such as Africa, contain a higher amount of melanin in their skin cells. So that is a great example of an adaptation of a cell.
Okay, so here we have how do cells survive, and this is just another content slide that we would present to the students so that they would have prior knowledge on the teacher experiment. So it states that cells survive through osmosis and just what osmosis is. So here I have conducted my own experiment, and I have documented the different stages of the experiment so that students are able to get snapshots of the process of osmosis. During this time, I would explain to the students what this process is and how it allows for the cells to get the nutrients it needs to survive, while relating it to the picture prompts I have taken. So as you can see, like our egg with the shell off represents the cell membrane, and then the yolk of the egg is the cell in itself and how water travels through, yet it doesn't affect the cell. So this is the rationale, and basically in the previous slides that we have conducted, the experiment demonstrates the scientific process of osmosis to expose students on how cells function and survive. We used a range of teaching strategies such as modeled teaching and work samples. This is another popular high impact teaching strategy. And this also gives a light introduction to the kind of experiments the year 6s will be conducting in the future regarding the function of the cells.
During the time of the presentation of content, I would get a series of pictures up on the PowerPoint or whatever we're using to present the content to the students, and we would give examples and non-examples of cells. So here we have a series of pictures that depict examples and non-examples of cells. We selected a wide range of objects, living organisms, to ensure that some were obvious and not so obvious to create discussion amongst the class. So during this time, I might, for example, show this picture of this animal here, and I would ask the students, do we think this animal might have cells? What kind of cells would the animal have? How do you know it has cells? And what might be the function of some of those cells? So like I talked about before, we might talk about the climate. Where does it live? Would that affect its cells? Would its direct environment affect the cells and the way that they function? So if we wanted to extend some of our students as well, we might ask, what living organism do you think this cell might belong to and why? So if we look at the diagram of the cell, there are some clues such as DNA. Where else is there? Some structures that don't actually exist in cells, such as plants or bacteria, and that only would live in a human cell. So we would get students to discuss this and justify their reasonings to extend them. And this is something that we haven't necessarily taught, but it's something that they might make a deduction about based off their own knowledge and understanding. So in our slides that include examples and non-examples, we have focused on the ideas of multiple exposures found in the high impact teaching strategies to promote discussion, familiarization, and test the students' current understanding of cells. Examples and non-examples is a strategy utilized to test the teacher's the students' grasp on the content and their own ability to justify and argue their understanding. The use of picture prompts has been utilized for our visual learners who struggle with a written word. We are ensuring to differentiate through the learning process so every student has the opportunity to learn.
All right, so this is our main activity. Sorry, technical difficulties, that is the hand there, but basically students are to choose one of the main cells in the human body and talk about it in the lesson. That has been talked about in the lesson, sorry. And then they are to make a model using the materials that have been provided and then label the model. So as you can see here, there's pictures of what the cell could look like and how it's labeled correctly. So during the explaining of the activity, we'll use explicit teaching where the teacher gives instructions, modeled work examples, and applies content knowledge to an activity. During this activity time, students who do require the extra assistance will use collaborative learning which is employed to cater to those students who do need the extra assistance. The teacher is gathering evidence of student knowledge through observation, questioning, and discussion. Example, what materials have you used to represent the cell membrane? Why? Why have you placed it there on the human body? And more. So this is known as assessment for learning. Then at the end of the activity, students' models are used as evidence of learning to determine if a student has met the success criteria, and this is known as assessment of learning. So in these two slides, we have outlined options from the lesson plan that extend and enable our students who need differentiation. This is an essential part of teaching and falls under the AITSL teaching practices as it is a requirement for all students. This allows us as teachers to give every student the best opportunity to learn as we want them all to meet the success criteria.
So here we have a set of reflection questions that we would present to the students after the main activity. Students would be asked to sit quietly at their tables and take some time to think and write the answers to these questions in their science workbook. So some of the questions we have here is, do you think you have achieved the success criteria, and how do you know? What do you need to keep practicing? And what would you wonder about cells? So getting into the inquiry part of our science as well. So what did the students actually want to know? What are they interested in? Self-assessment is an important piece of evidence for assessment as learning. Students will determine themselves if they have achieved the success criteria. This is important as it gives students the opportunity to evaluate their own performance so they can improve and set goals for new learning. This may also be an opportunity for a teacher to receive feedback from their students if a particular student was not connecting with an idea. It's important as a teacher to reflect to come up with new ways to help that student connect with the content.